Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So, in the Catabolism of Carbon Skeletons of Amino Acid lecture series, so here in this particular lecture, I will be going over amino acids that produce Succinyl-CoA as one of the TCA cycle intermediate. Let's get into the details. So, amino acids that produce Succinyl-CoA, because Succinyl-CoA is one of the TCA cycle intermediate and which can eventually go into glucose formation. So, those amino acids are Methionine, Valine, Isoleucine and Threonine. Now, let's get into see metabolism of methionine and how the carbons of methionine will be converted to Succinyl-CoA and how Succinyl-CoA goes into gluconeogenesis process. So, as it is shown here in the figure, methionine is converted into S-adenosyl-methionine by using ATPs, and S-adenosyl-methionine will undergo methylation reactions where a methyl group is donated to a variety of methyl group acceptors in the methylation process. I am going to give you the list of reactions where S-adenosyl-methionine, that is SAM, is used as a methyl group donor in a methylation process. Once the methyl group is donated by S-adenosyl-methionine, so S-adenosyl-methionine is converted into S-adenosyl-homocysteine. And now the S-adenosyl-homocysteine undergoes hydrolase reaction where adenosine is released and homocysteine comes out of the reaction. Now, the homocysteine gets a carbon skeleton from serine to make cystathionine. Now, the cystathionine will be converted into cysteine and alpha-ketobutyrate, and of course, there will be release of ammonium ion. Now, what happened here is the sulfur present in methionine, it will be given to cysteine, and the carbons of methionine will be going into alpha-ketobutyrate formation. Let me just write down and track that sulfur and carbons. So, I am going to write a sulfur here for methionine. So, we are going to concentrate on the sulfur and also the carbons. Okay, carbon I am going to write in blue color. Okay, so carbons are in blue color and sulfur in red color. So, you know methionine is a sulfur containing amino acid. So, methionine is converted to S-adenosyl-methionine. So, we have carbons in the carbons of methionine in S-adenosyl-methionine and we have a sulfur also there. And S-adenosyl-homocysteine also has the carbons of methionine and sulfur of methionine. So, it continues there. So, carbons and sulfur still continued in the form of S-adenosyl-homocysteine. Similarly, homocysteine also has carbon and sulfur. So, it is continued. Okay. Now, homocysteine is converted to cystathionine. It is just a condensation of homocysteine with serine. Serine is coming in now and making cystathionine. So, carbons of homocysteine, sulfur of homocysteine, now the carbon, nitrogen of serine, everything is there in the cystathionine now. So, I will just continue to write sulfur is there in coming from methionine is there in. So, carbons are there coming from methionine, sulfur is also there here in cystathionine. So, everything is retained. Now, let us see what happens. Cystathionine, it breaks into cysteine and alpha-ketobutyrate. Now, something changes. So, here what happens is when the cysteine is forming and alpha-ketobutyrate is released, so the carbons will be there in alpha-ketobutyrate. So, carbons of methionine, you are going to find that in alpha-ketobutyrate, whereas the sulfur of methionine, it is there in cysteine. So, sulfur is there in cysteine now. So, that means methionine is the donor of sulfur that is present in cysteine. What about the carbons of cysteine? Carbons of cysteine are actually coming from serine. Let me write that also here. So I am going to color carbons of serine with a violet color here. So carbons of serine, they are present in cystothionine after it condenses with homocysteine, and then the carbons of serine is found in the cysteine. So cysteine has carbons coming from serine and sulfur coming from methionine. So carbons are coming from serine, make sure you remember this, and sulfur is actually coming from methionine in the cysteine. Okay, and that's how the cysteine is made here. Cysteine is a semi-essential amino acid. It gets carbons from the serine and sulfur from methionine. So the sulfur of methionine is transferred all the way to cysteine. So this is what is the trans-sulfuration pathway of methionine. So methionine, it has transferred its sulfur from methionine all the way to cysteine. That is a trans-sulfuration process. And also the carbons of methionine now, they are present in alpha-ketobutyrate. Let's see what happens to alpha-ketobutyrate. So this alpha-ketobutyrate will undergo further metabolic processes and ultimately converted to propionyl-CoA, and propionyl-CoA will be converted to Succinyl-CoA. We are going to see that in the subsequent slides. And this is what happens to the carbons of methionine. So the carbon skeleton of methionine, make sure you remember, while it undergoes all this methylation reaction, acetylmethionine, acetylhomocysteine, homocysteine, cystothionine, cysteine and alpha-ketobutyrate, and alpha-ketobutyrate carries the carbons of methionine and become propionyl-CoA, which becomes Succinyl-CoA. Succinyl-CoA can go into gluconeogenesis. That's how the carbons of methionine can form propionyl-CoA, becoming Succinyl-CoA and glucose. Okay, so sulfur is transferred to cysteine. And now we have already seen in my previous lecture what will happen to the cysteine. Okay, carbons of cysteine and sulfur of cysteine when it undergoes catabolism. Alright, now see here in this particular metabolic pathway. So we have seen trans-sulfuration process. So I am going to track that sulfur, methionine sulfur. It is present in homocysteine now, and homocysteine to cystothionine and cystothionine to cysteine. This is how the sulfur is transferred from all the way from methionine to cysteine. That's the trans-sulfuration pathway. This is called as trans-sulfuration pathway, where sulfur is transferred from methionine all the way to cysteine. So the sulfur is donated from methionine, that is we take. So the sulfur present in cysteine is donated by methionine. Now in this trans-sulfuration pathway, I would like to draw your attention to one of the coenzyme that is required in the trans-sulfuration pathway. As you see here, conversion of homocysteine into cystothionine, you need a pyridoxal phosphate, PLP, as the coenzyme, and you know PLP is an active form of vitamin B6. And conversion of cystothionine into cysteine and alpha-ketobutyrate also needs PLP as the coenzyme. So in the trans-sulfuration pathway, there is a requirement of pyridoxal phosphate. Okay, so the enzyme that catalyzes the two enzymes, important ones, that are catalyzing the conversion of homocysteine into cysteine and alpha-ketobutyrate in the trans-sulfuration process is, the first reaction is catalyzed by cystothionine beta synthase enzyme. So the name of the enzyme is cystothionine beta synthase. Cystothionine beta synthase catalyzes the conversion of homocysteine into cystothionine. And the second enzyme in this pathway, conversion of cystothionine into alpha-ketobutyrate and cysteine, is a cystothionase enzyme. Cystothionase. So cystothionine beta synthase and cystothionase enzyme, both of them are participating in trans-sulfuration pathway, and both of them they need pyridoxal phosphate as the coenzyme. Imagine if there is a mutation in a gene coding for cystathionine beta synthase enzyme. So what will happen? So the reaction that is converting homocysteine into cystathionine will decrease, and that will lead to elevation of homocysteine, right? And that's what causes type 1 homocysteinemia. Type 1 homocysteinemia. Type 1 homocysteinemia is because of deficiency of enzyme cystathionine beta synthase or defect or decreased activity of cystathionine beta synthase. It can also be partly because of pyridoxal phosphate deficiency. So the deficiency of cystathionine beta synthase enzyme will lead to decrease in the conversion of homocysteine into cystathionine. It can also be because of deficiency of pyridoxal phosphate as the coenzyme. And what if there is a decreased activity of other enzyme, that is cystathionase enzyme? That will affect the second reaction here, that is conversion of cystathionine into alpha-ketobutyrate and cysteine. So that reaction will decrease and leads to elevation of cystathionine now. And that causes a disease called cystathionuria. So these are the two disorders that are in transsulfuration pathway. So I told you what is transsulfuration pathway there. So two important enzymes in terms of clinical application. One is conversion of homocysteine into cystathionine, cystathionine beta synthase enzyme leading to type 1 homocysteinemia. Conversion of cystathionine into alpha-ketobutyrate and cysteine, deficiency of cystathionase enzyme leading to cystathionuria. Now let's continue to see what will happen to alpha-ketobutyrate that is made in this particular process of conversion of methionine all the way into cysteine and alpha-ketobutyrate. So the alpha-ketobutyrate, as it is shown, will continue and become propionyl-CoA after decarboxylation process, and the propionyl-CoA, which is a 3-carbon molecule, will be converted into methylmalonyl-CoA by propionyl-CoA carboxylase. I have already explained you on conversion of propionyl-CoA to methylmalonyl-CoA into by propionyl-CoA carboxylase in oxidation of odd number carbon fatty acids. In my previous lecture, I have detailedly explained conversion of propionyl-CoA into methylmalonyl-CoA. Enzyme is propionyl-CoA carboxylase. Which needs a biotin as the coenzyme. Now methylmalonyl-CoA D form converted to L form, and then L-methylmalonyl-CoA is converted to Succinyl-CoA by methylmalonyl-CoA mutase, methylmalonyl-CoA mutase enzyme, and that needs vitamin B12, especially 𝗑𝐿𝐳-𝐸𝐻𝐼𝑛𝑜syl-cobalamin as the coenzyme. Once you get succinyl-CoA, succinyl-CoA gets into TCA cycle becoming succinate, succinate to fumarate, fumarate to malate, malate gets out of mitochondria into the cytoplasm, and we can go into glucose formation. That's how ultimately the carbons of methionine will form alpha-ketobutyrate, and alpha-ketobutyrate will form propionyl-CoA, propionyl-CoA all the way converted to succinyl-CoA, and succinyl-CoA go into gluconeogenesis. And while this process of conversion goes on from alpha-ketobutyrate all the way to propionyl-CoA, succinyl-CoA to glucose, so at least in this particular pathway, so there are two enzymes that can have a clinical application. One is propionyl-CoA carboxylase, conversion of propionyl-CoA into methylmalonyl-CoA. Let me take this out here, whatever the things that I have written. So conversion of propionyl-CoA into methylmalonyl-CoA done by propionyl-CoA carboxylase, and which needs biotin. So deficiency or defect in this reaction lead to elevation of propionyl-CoA, and that propionyl-CoA will lead to elevation of propionic acid in the blood and appearing in the urine. That causes propionic acidemia. Propionic acidemia. That's the disease. Now the second one is, what if there is a defect in the conversion of methylmalonyl-CoA into succinyl-CoA done by methylmalonyl-CoA mutase enzyme, which needs vitamin B12 as the coenzyme. So defect in the enzyme or decreased availability of vitamin B12 will lead to elevation of methylmalonyl-CoA, causing methylmalonic acidemia. So we have two diseases in the conversion of alpha-ketobutyrate all the way to succinyl-CoA. One is propionic acidemia and methylmalonic acidemia, and you must be aware of that. And one more point I want you to remember while we are in this pathway, which is kind of important, is what are the amino acids that makes propionyl-CoA as their metabolic fate while the carbon skeleton is undergoing oxidation. So it is, you need to remember the names: threonine, amino acid threonine becoming alpha-ketobutyrate and becoming propionyl-CoA. So carbon skeleton oxidation of threonine will give propionyl-CoA. That's one thing. Second, isoleucine. Carbon skeleton of isoleucine upon oxidation gives propionyl-CoA. Third, valine. Carbon skeleton of oxidation of valine will give propionyl-CoA. And of course, methionine. Oxidation of carbon skeleton of methionine is going to give propionyl-CoA. So, 4 amino acids carbon skeleton upon oxidation will give propionyl-CoA as their metabolic intermediate. Okay, those 4 amino acids are methionine, threonine, isoleucine and valine. And also in my previous lectures, many times I have mentioned all the sources of propionyl-CoA in our body. What all the different ways that propionyl-CoA can come in. And the mnemonic to remember that was, I told Bob needs vitamin M. So the last part of this mnemonic, vitamin M, is there here in this slide. That V for valine, I for isoleucine, T for threonine and M for methionine. All of them will give propionyl-CoA. What is Bob? It is oxidation of branched chain amino acid. So the oxidation of branched chain fatty acid, oxidation of odd number carbon fatty acid and bile acid synthesis. That's where other propionyl-CoAs will come. Alright, so this is what is about how carbons of methionine will go all the way to alpha-ketobutyrate and eventually forming succinyl-CoA. So the amino acid that produces succinyl-CoA is a methionine here. Okay, and of course threonine, isoleucine, valine via propionyl-CoA will also make a succinyl-CoA as a tertiary cycle intermediate. Alright, now while I was explaining a trans-sulfuration pathway, I also want you to go over something called as methylation pathway of methionine. So I will briefly explain that here, and then in the later slides I will go over little more in detail. So as you see here, homocysteine going down into cystathionine and cystathionine into cysteine, and that's where trans-sulfuration pathway was going on. Okay, now if you go back to formation of methionine from homocysteine, like homocysteine has another fate, it can go back into methionine side. And during that process of conversion of homocysteine back into methionine, there is a need of methyl group. Because you know methyl group is donated when 𝐒𝐮𝐬𝐜𝐫𝐢𝐛𝐿 is converted to 𝐒-homocysteine, methionine is given out. So homocysteine back into methionine, so the methyl group has to be given back. That is what is called as methylation pathway. So in the methylation pathway of conversion of homocysteine into methionine, there is a requirement of 2 vitamins. One is cobalamin in the form of methylcobalamin, that's a type of cobalamin. Other is N5-methyl tetrahydrofolate. These are the two vitamins that are necessary in the methylation pathway of conversion of homocysteine into methionine. Remember that. Alright, so from the previous figure, you can see conversion of homocysteine from methionine all the way into alpha ketobutyrate. It undergoes oxidative decarboxylation to form propionyl-CoA, and propionyl-CoA is converted to succinyl-CoA. And the carbon skeleton for cysteine synthesis is provided by serine, which I already told you, and the sulfur is provided by methionine in a transsulfuration pathway. Now, whenever serine whenever cysteine levels elevated in the cell, the cysteine will have a feedback negative effect on enzyme called cystathionine beta synthase, which is involved in the conversion of homocysteine into cystathionine. Alright, so these are some of the points which are a recap from the previous figure that I have explained you. Now let me explain you briefly methylation pathway. So homocysteine is converted into methionine upon taking methyl group from vitamin B12, that is in the form of methylcobalamin. Methylcobalamin is a type of vitamin B12. So the methylcobalamin is going to give a methyl group to homocysteine. That's when homocysteine is converted into methionine. During this process, what will happen? The cobalamin, methylcobalamin will be converted into just cobalamin. Let me write down here. So what I am saying here is methylcobalamin, I will write it as CH3B12. Here it is just simply written as B12, but it is actually coming in as CH3B12, that is methylcobalamin, will go out of the reaction as just cobalamin B12. And this just cobalamin will be converted back into methylcobalamin regeneration by using a methyl group that is coming from N5-methyl tetrahydrofolate. Let me write it on the side here so that you understand it properly. Homocysteine is converted into methionine. That's what is the methylation pathway. Methyl group is given. How? CH3B12, that is methylcobalamin, is the CH3B12, is giving that methyl group to methionine and going out of this reaction as just B12, cobalamin, just cobalamin. Methylcobalamin is giving the methyl group, that's the methylation process done by methionine synthase enzyme. Methionine synthase, it's a transferase enzyme. It is going to transfer methyl group, homocysteine methyl transferase, that's the another name of methionine synthase. So CH3B12 giving methyl group, going out as B12, and that B12 has to be regenerated back into CH3B12 just to maintain the concentration of methylcobalamin in the cell so that homocysteine conversion into methionine continues. So we'll have to recycle it, regenerate, right? In the regeneration process, something else has to give methyl group to cobalamin so that cobalamin is converted back into methylcobalamin, and that is given by N5, that is 5th nitrogen of folic acid is attached with the methyl group N5 methyl, that is CH3 tetrahydrofolate. N5 methyl tetrahydrofolate gives that methyl group to cobalamin and go out as just tetrahydrofolate. And this tetrahydrofolate can go into different tetrahydrofolate formation, including reformation of tetrahydrofolate back into N5-methyl tetrahydrofolate. So overall, what happened here? Methylcobalamin is giving methyl group to homocysteine to make it as methionine. That's a methylation process, methylation pathway for homocysteine into methionine. And then during this process, when the methyl group is given by cobalamin, it will just go out of the reaction as just cobalamin. That cobalamin has to be converted back into methylcobalamin. So the methyl group donor at that time is N5-methyl tetrahydrofolate. That is what is shown here, in a brief way. And now, after donating the methyl group by N5-methyl tetrahydrofolate, It is just going out of the reaction as just tetrahydrofolate. And that tetrahydrofolate can go into different tetrahydrofolate formation. Basically, it enters into tetrahydrofolate pool. There are different types of tetrahydrofolates: N5-methyl tetrahydrofolate, N5-N10 methylene tetrahydrofolate, N5-N10 methyl tetrahydrofolate, formyl tetrahydrofolate, different types. Okay. So, one of the things that is shown here in the figure is the tetrahydrofolate, how it can regenerate back into N5-methyl tetrahydrofolate. The intermediate there is N5-N10 methylene tetrahydrofolate. So, tetrahydrofolate can form N5-N10 methylene tetrahydrofolate. That's where the glycine metabolism comes in. You know, in the previous lecture, I told you metabolism of glycine to serine, where like N5-N10 methylene tetrahydrofolate comes in, or when the glycine breaks by glycine cleavage enzyme, there will be release of N5-N10 methylene tetrahydrofolate, right? And that N5-N10 methylene tetrahydrofolate can be converted back into N5-methyl tetrahydrofolate involving a vitamin B12 riboflavin. Okay, so like this, all these conversions will go on, and you got to remember there that particular aspect, because any issues here, any defects here can lead to elevation of homocysteine. Why? Because what if cobalamin is deficient? What if cobalamin is not available? Cobalamin deficiency, vitamin B12 deficiency. That will affect methylation pathway in methionine metabolism. So that means there will be elevation of homocysteine, homocysteinemia. It can lead to homocysteinemia, called type 2 homocysteinemia. What if there is a deficiency of folic acid itself? So, N5-methyl tetrahydrofolate is not available sufficiently. That means regeneration of cobalamin back into methylcobalamin do not go on. That can also lead to elevation of homocysteine. What if there is a defect in the process of conversion of N5-N10 methylene tetrahydrofolate? There is something called a DHFR, dihydrofolate reductase enzyme. Defect or deficiency in dihydrofolate reductase enzyme will lead to decreased availability of N5-N10 methylene tetrahydrofolate. That can also lead to homocysteinemia. So, that is why you got to remember these details, some of the coenzymes that are coming into the conversion of homocysteine into methionine. So, there is a role of cobalamin and folic acid derivatives in the conversion of homocysteine into methionine. Remember that. Okay. So, that's what I wanted to explain in methylation pathway. So, the other fate of homocysteine is to convert back into methionine. So, this can happen in two ways. First, reaction requires both folate as methyl tetrahydrofolate and vitamin B12 as methylcobalamin catalyzed by methionine synthase. The second involves the transfer of methyl group from betaine, a metabolite of choline, as you see here in the figure here. So, the other part that I missed to explain in the previous explanation, as you see here, homocysteine can also go into methionine, although it is a less preferred or less frequent pathway of conversion of homocysteine into methionine. So, the betaine is the methyl group donor, and betaine comes from choline. And you know, choline, it is coming from phosphatidylcholine. You know, membrane phospholipid, phosphatidylcholine. Phosphatidylcholine is lecithin. Other name for phosphatidylcholine is lecithin. Lecithin is the one that donates choline, and also choline can come from diet. So, this is a membrane lipid. Phosphatidylcholine is a membrane lipid, or it can come from diet like egg yolk as a choline. So, whenever there is a deficiency of cobalamin, deficiency of folic acid, that leads to increased accumulation of homocysteine in the tissue or cell, and some of that homocysteine can still be converted into methionine by using betaine. At that time, there is a consumption of choline, because to make more and more betaine, choline is consumed. And this choline it has to come from phosphatidylcholine. So the membrane phosphatidylcholine is kind of it means we got to degrade that membrane lipid, that is phosphatidylcholine, to provide choline. If it is not coming from the diet, it has to come from the membrane. So membrane has to undergo degradation there. That can lead to altered permeability in the membrane. Remember that. So homocysteinemia can affect neuronal changes. It will affect so many membrane changes because it is degrading the membrane for phosphatidylcholine to provide choline. And also note that choline is also necessary for formation of acetylcholine. Acetylcholine is a neurotransmitter, parasympathetic neurotransmitter. It has to come from choline. So that can also be affected because if it is just directed towards betaine formation. So things like this can be connected here. Okay. So that's about how homocysteine is converted into methionine. There are two ways to do that, as I am explaining here. One way is using folate and methylcobalamin, methyl tetrahydrofolate and methylcobalamin by methionine synthase. Other way is using methyl group from betaine, that is coming from choline. Alright. Now let's look at the S-adenosyl-methionine and transmethylation reactions. You got to make sure know some of these details here because important concept. Many questions can appear here. So because one of the intermediate in the conversion of methionine into S-adenosyl-methionine, it is formation, because one of the intermediate in the formation of methionine into homocysteine and then going into cysteine and alpha-ketobutyrate, we have seen the formation of S-adenosyl-methionine, where ATP gets in and only the adenosine is donated, 3 phosphates are going out. So S-adenosyl-methionine, it will participate in a methylation reaction because the precursors which will accept the methyl group enters into the reaction, uses the methyl group from S-adenosyl-methionine and they will go out of the reaction as methylated product and S-adenosyl homocysteine will be released. Okay. Now let's see what all the reactions that uses SAM as the methyl group donor. So conversion of norepinephrine into epinephrine in the neurotransmitter synthesis, especially in the adrenal medulla and some of the sympathetic neurons in the central nervous system, uses a methyl group that come from S-adenosyl-methionine. And conversion of norepinephrine into epinephrine, it will be done by an enzyme phenylethylamine N-methyltransferase. Okay. And then the conversion of guanidoacetate into creatine, and that uses S-adenosyl-methionine, methyltransferase reaction there. Creatine it can be converted into creatine phosphate and it will be stored in the skeletal muscle, the cardiac tissues, and it is a storage form of energy. Whenever a person is relaxing, when more ATPs are available and creatine is made and it will be phosphorylated to creatine phosphate, and whenever ATPs are needed, creatine phosphate breaks and phosphorylate ADP to make ATP and molecule is released as creatinine and that will be secreted out of the kidney into the urine. So it will find its way into the urine, creatinine, right. So and then nucleotides like DNA methylation process uses a SAM as the methyl group donor. So methylated nucleotides. And then phosphatidylethanolamine converting into phosphatidylcholine uses S-adenosyl-methionine. So phosphatidylcholine, that is the membrane phospholipid, which is the donor of choline in the conversion of homocysteine into methionine, or in so many like acetylcholine synthesis. So during that time also you need SAM as the methyl group donor. And then conversion of acetyl serotonin into melatonin. Melatonin is synthesized in the pineal gland and it is maintaining a circadian rhythm. So melatonin synthesis needs SAM. So these are some of the reactions where SAM is used as the methyl group donor, and make sure you remember these reactions, very important. That is formation of epinephrine, creatine, methylated nucleotides, phosphatidylcholine and melatonin formation. They all need SAM as the methyl group donor. And now while I am here, so let me just briefly introduce you, although I have explained in the previous slide, just to show it in a different way. Conversion of homocysteine into methionine. Here the methylcobalamin, type of cobalamin, is giving that methyl group and going out of the reaction as just cobalamin there. Okay, so methyl group is given out, and now the cobalamin has to be converted back into methylcobalamin so that the cobalamin levels are sufficiently methylcobalamin levels are sufficiently available to convert homocysteine into methionine, and that will be done by N5-methyl tetrahydrofolate, which is donating its methyl group, going out as tetrahydrofolate, and tetrahydrofolate can go into different tetrahydrofolate formation. Now there is a concept called as folate trap, as you see here. What is folate trap? Folate trap is in the deficiency of cobalamin. Whenever there is a cobalamin is deficient, the folate in the cell, it will be trapped as N5-methyl tetrahydrofolate, thereby it will lead to functional folate deficiency. Let me explain that concept to you. Now what we will do is, we are going to decrease cobalamin, because consider that person has got cobalamin deficiency, so I am going to decrease cobalamin levels here. Okay, cobalamin is decreased, cobalamin deficiency. During cobalamin deficiency, what happens? So we don't have much cobalamin now, so the conversion of N5-methyl tetrahydrofolate into just tetrahydrofolate, while B12 conversion into methyl B12 will decrease. So what will happen? So there will be accumulation of N5-methyl, this is N5-methyl tetrahydrofolate, that's how we need to say that. N5-methyl tetrahydrofolate simply accumulates in the cell. And what happens to the tetrahydrofolates, free tetrahydrofolate? Free tetrahydrofolate levels will decrease in the cell. And these tetrahydrofolates are free tetrahydrofolate is necessary to go into different direction, different tetrahydrofolate formation, and that is not going on. So overall what happened here, most of the tetrahydrofolate in the cell, it is just trapped as N5-methyl tetrahydrofolate. So this happens because of cobalamin deficiency, because this is the only reaction here which will convert N5-methyl tetrahydrofolate into tetrahydrofolate, freeing the tetrahydrofolate to go into different direction. So here tetrahydrofolate is trapped inside the cell as N5-methyl tetrahydrofolate. Although tetrahydrofolate is there, but it is not free. So that leads to functional folate deficiency. Functional folate deficiency means folate is there, but it's not available for the function. Okay, and when do you see functional folate deficiency? Functional folate deficiency is seen whenever there is a cobalamin deficiency. That's the connection here. Cobalamin is connected with the folate. So cobalamin deficiency will lead to functional folate deficiency. That means whenever there is a cobalamin deficiency, the signs and symptoms of cobalamin deficiency includes folate deficiency. That's what I want you to know. Okay. So in the vitamin lecture, I have explained folate deficiency will lead to megaloblastic anemia. Folate deficiency causes megaloblastic anemia. That's the clinical feature of folate deficiency. Now what about cobalamin deficiency? Cobalamin deficiency also show megaloblastic anemia. Along with megaloblastic anemia, cobalamin deficiency will show neurological signs, especially subacute combined degeneration of spinal cord. So that is one of the neurological issue you are going to see in cobalamin deficiency, and also patients will show elevated methyl malonate or methylmalonic acidemia. So, in cobalamin deficiency, you are going to see neurological issues and metabolic acidosis because of methylmalonic acidemia. Along with that, we are going to see megaloblastic anemia. So, the megaloblastic anemia component of cobalamin deficiency is because of folate trap. Why? Because in folate trap is what? Cobalamin deficiency leads to functional folate deficiency. So, all the signs of folate deficiency you also see in cobalamin deficiency. That is why even like before conception or like during early weeks of pregnancy, if the folic acid levels are low, it will lead to miscarriages, neural tube defects, infertility thing. All that is although related to folic acid, but you know cobalamin deficiency leads to functional folate deficiency. That's why neural tube defects can be seen both in folic acid deficiency and also in cobalamin deficiency. Remember that. Okay. So, cobalamin and folic acid deficiency can lead to infertility, it can lead to frequent miscarriages, it can also lead to neural tube defect in the developing fetus inside. Okay. Remember the concept, it is very important. Alright. Now, let us move on to see other amino acids that can make Succinyl-CoA as their metabolic intermediate, especially coming from the carbon skeleton. Next slide. So, what you see here is branched chain amino acids, valine, isoleucine and leucine. Especially look at valine and isoleucine. So, valine and isoleucine, they will make propionyl-CoA, and propionyl-CoA will go into Succinyl-CoA formation, which I have already explained you. So, both valine and isoleucine, even the leucine one, which we have already seen in the amino acids that make acetyl-CoA or acetoacetate. In that particular lecture, I have explained leucine. Now, valine and isoleucine undergoes transamination and oxidative decarboxylation to decarboxylation and deamination process. So, valine and isoleucine, so they will undergo transamination and oxidative decarboxylation to make alpha-ketoisovalerate, alpha-keto-beta-methylvalerate. Upon further decarboxylation, they will undergo isobutyryl-CoA and methyl-butyryl-CoA, and they will undergo further oxidative reactions to make propionyl-CoA, and propionyl-CoA further converted into Succinyl-CoA. So, like this, oxidation of valine and isoleucine will make propionyl-CoA and propionyl-CoA making Succinyl-CoA, and that is a glucogenic amino acid. And know that there can be a defect in the oxidative decarboxylation process. And note that there can be a defect in the transamination, oxidative deamination process, or there can be accumulation of certain things in the metabolism of valine, isoleucine, leucine, and that can be a clinical application. One of the things that I would like to mention here before I go over the disease in detail in my next lecture is a defect in an enzyme that is catalyzing oxidative decarboxylation of branched chain amino acids. Name of the enzyme is branched chain alpha keto acid dehydrogenase. It will lead to elevation of the keto derivatives of all three branched chain amino acids, that is alpha keto isovalerate, alpha keto beta methylvalerate and alpha keto 𝐒𝐼𝐳-CoA. Valine gives alpha keto isovalerate, is there in the name. Isoleucine gives alpha keto beta methylvalerate, leucine gives alpha keto 𝐒𝐼𝐳-CoA. They all will accumulate, and once they accumulate, they will lead to accumulation of valine, isoleucine, leucine in the tissues and appearing in the blood, because transamination is a reversible reaction. So, the defect in alpha keto acid dehydrogenase complex will give rise to a disease called maple syrup urine disease. It is written here, maple syrup urine disease. So, block in this particular reaction. Okay. So, that we need to know the enzyme name there, which will lead to maple syrup urine disease. So that's about the amino acids that make or that produce succinyl-CoA as their carbon skeleton is undergoing oxidation. Thank you.